Today we are learning how the mechanics behind heredity were discovered. We want to learn this so we can understand the patterns of inheritance of traits from one generation to the next. And our success criteria for today is being able to explain who Mendel was and why he experimented with pea plants. So, Gregor Mendel was an Augustine friar born in the Austrian Empire, which today he would have been born in the modern day Czech Republic. Mendel was not recognized as a great scientist in his time. He was a scientist, he considered himself to be a scientist, but his work was so groundbreaking and cutting edge that in the 19th century, when he was working, nobody else could understand what he was doing. And Mendel very famously said to some of his friends that my time will come, because he knew he was right and he knew that he had landed on something very important, even if no one at the time could understand it. And he was right. His time did come. His work was rediscovered in the year 1900 by three different scientists independently in about a 2-month period. And all three scientists were able to independently replicate Mendel's work, to repeat his experiments, and they got the same results. And they all realized that there was something very deeply profound about what Mendel discovered. And to all of their credit, they all gave Mendel the credit as being the founder of heredity, even though they didn't learn their work directly from him and they made some conclusions on their own. They recognized and acknowledged that he was the first one to discover this, and that without his work, they would not have been able to complete theirs. So today, Mendel is known as the father of genetics because he really did blow open the entire field. And the interesting thing is he did this with no understanding of DNA. He was trained as a botanist and as a mathematician, and he was the first person to really give a solid application of statistics to biology. He was able to use statistics and probability to extrapolate several key ideas that are essential to the field of genetics. What Mendel was looking for was he wanted to understand the patterns of transmission of traits from one generation to the next. How did traits get passed down from parents to children? And he was very successful because he was a very thorough bookkeeper. He tracked his results very systematically. And then because he was so good at mathematics, he was able to give a very clear and insightful analysis of his data. So what Mendel used for his experiments were pea plants. They were not his first choice, but he tried several different organisms and eventually settled on the peas. He picked these because they were cheap and easily available. They were very easy to grow, which meant he could raise a lot of them at the same time, and they matured very quickly, so he could go through generations of pea plants very fast. The pea plants also had the added benefit of Mendel was able to control their reproduction. If you look at those flowers on the right-hand side diagram, you'll notice that the fronts of them are very closed off. So what that meant was that it would be difficult for them to be fertilized by another plant unless Mendel did it directly. They were also able to self-pollinate. So Mendel could actually take one plant and breed it with itself, which we'll see in a couple of lectures is going to be very important. So all of that meant that Mendel could track exactly which plant was breeding with which other plant. Mendel also got a little bit lucky, although he picked these peas for this reason as well, in that they had very clear and distinct traits. And genetically, what that means is that the genetics behind these traits are very simple. For Mendel's purposes, it also meant it was very, very easy for him to track these traits. So he would be able to see what the patterns of inheritance from one generation to the next would be with minimal work. Like I said, he did get a little bit lucky here because he didn't know anything about DNA, and he picked a very, very good organism to do a simple first sample.